for a little bit, so we'll go ahead and get started. So welcome everyone, and thank you very much for coming to the Working Group on Pediatric Gene Therapy and Medical Ethics, which we finally call PGTME, our third annual lunchtime lecture series. My name is Alison Bateman-House. I'm an assistant professor of medical ethics at NYU Grossman School of Medicine, and I'm co-chair of PGTME along with Dr. Lesha Shah, who will be moderating a session later this week. Our panelists will be introducing themselves shortly. I just have a few introductory remarks, mainly that we're so grateful that you're all here for our third year of hosting this event. Every year it's getting bigger and better, and we're delighted to have you be a part of this with us. And we're always impressed by and inspired by the diversity of people who participate in this event and the absolutely wonderful questions they submit. So please, at any point today that you have a question, submit it through the chat function, and I'll get to as many of them as possible. And we're going to be reserving 10 minutes for those questions specifically. But again, if you get them in early, I'll try to work them into the conversation as early as I can. If you're unfamiliar with PGTME, you can learn more about our group, our members, and our activities at our website. Kara Hunt, who is the project manager, will put the link in the chat for you. And you can sign up for our quarterly newsletter that we put out that will help you stay abreast of all of our work. So we're very excited to hear from a cast of amazing speakers today about the state and future of the field of pediatric gene therapy research. And we have interesting topics all week. I would especially like to recommend to you Friday's panel discussion. It is with a former pediatric gene therapy research participant, as well as parents and siblings of other pediatric patients, some of whom have and some of whom have not opted to participate in pediatric gene therapy research. And please remember that if the 12 to 1 p.m. slot doesn't work for you, as long as you register, we will send you a link to the recording afterwards, and a posted recording with transcript will be available online on YouTube. But that said, we do hope that you participate live whenever possible so that you can participate in the Q&A. So that's what's happening this week. But as for today, we're going to, again, as I said, have brief introductions by our panelists. I've asked them to keep them brief because their information, their titles and whatnot is available on the Eventbrite page where you signed up to attend this. And Kara just put the link to the speaker bios in the chat to make it even easier. And so that is what we're going to do. Without further ado, I'm going to hand it off to the panelists to introduce themselves and their work. So can we start with Dr. Radina Klepek?
Sure. Hi, nice to meet everyone today. I'm Louise Radina Klepek. I'm a PhD scientist, spent the majority of my career in gene therapy research, primarily for muscular dystrophies. I was at Nationwide Children's in academia, where I developed several gene therapies for muscular dystrophies. And then about 4 years ago, I moved to Sarepta Therapeutics, where I now head R&D.
Thank you. Dr. Brooks?
Yes, hi, I'm PJ Brooks. I'm the acting director of the Division of Rare Disease Research Innovation at NCATS, part of the NIH, PhD neuroscientist by training. And I originally kind of got motivated in this field from working on a rare disease and seeing firsthand the challenges and trying to think about bringing a gene therapy into the clinic for a very, very rare disease of no commercial interest. And in my role now at NCATS and NIH, I'm involved in the oversight of several different efforts on gene-targeted therapies, including gene therapy and gene editing.
Thank you, PJ. And last but certainly not least, Dr. Flotte.
Yeah, hi, Terry Flotte. I'm a pediatrician and pediatric pulmonologist, and I've been working primarily with AAV gene therapies in clinical trials over quite a few years. I also have had a number of different leadership roles in the American Society of Gene Cell Therapy and editor-in-chief at Human Gene Therapy, which is the European Society's official journal. So try to stay connected to those official societies as well.
So you can see we have a range of experts for you today from academia, industry, NIH. You'll note that there are two other names on your screen that have their cameras off. That is Kara Hunt, my very impressive and skilled program manager for PGTME, who's helping running things behind the screen scenes, and also Aiden, who is also equally talented and equally essential to running things behind the scenes. So with that, we'll get started. And again, I just want to encourage you to drop things in the chat as you have questions or need clarifications or whatnot, and we'll try to handle as many of them as possible. So I'm just going to throw this one out to the panel, and whoever is interested can grab it and run. I had a just very basic question. You know, we just saw the 5th gene therapy product approved by FDA last week. There's been, you know, exciting advances in the field. There's certainly been a massive increase in research, both in pediatric populations and adult indications. And I was just wondering what people see as sort of like the most exciting or most, you know, really interesting thing that's been happening in this field in the last year or so. What is making you excited about working in this area?
All right, I'll jump in.
Go ahead,
Oh,
Terry.
I was just going to say, I think you tapped into it, Alison, saying that all these approvals for this range of diseases, including some very rare diseases, have been very exciting. And the approval for the Unicure factor 9 was a big one, but I think just in the previous months we had an approval for the early cerebral ALD that came out from Bluebird. We had the congenital AADC deficiency in Europe from PTC, also a hemophilia A product approved in Europe. And, there's just so many of these genetic disease gene therapies that are hitting the approval stage, which is really just the tip of the iceberg of all the different things that are in trials. So that's very exciting to me.
Thanks, Terry. PJ, you were trying to get in?
Yeah, I certainly echo that. I think it's wonderful to see these approvals based in Europe and the US. I think we're starting to see gene therapy fulfilling the promise that we all thought it would have. When I think of what I'm most excited about, it's a bit more looking into the future, but I think particular in the area of genome editing and more specifically these what I would think of as novel types of genome editors, such as the base editor, as the prime editor. These would be single biologics that could in principle treat large numbers of monogenic diseases. And I say this with quotation marks, the only thing you need to do to change from one to another or different gene targets is to change the sequence of the guide RNA. And I'm particularly excited about therapeutic platforms. AAV is certainly a therapeutic platform that we're trying to exploit in various ways through some of the programs that I'll talk about later. But I certainly think that genome editing, particularly the base editor and prime editors, are very exciting technologies. And the fact that they're already getting into the clinic is something I'm very excited about.
I'll just echo a lot of what was said. I think there's a tremendous amount of reason to be excited about the state of the field. I think we always knew the promise was there of what gene therapy can accomplish and do. And I think whatever challenges that the field has faced, we've found ways to overcome them and continue to do that. So I think there's a lot of optimism. I think the recent approvals have been fantastic. I think you'll see a lot of applications and hopefully approvals over the next year. We recently submitted a BLA as well. So I think there's just for me, I think the tide has turned and there's a tremendous amount of optimism, and what we can do for patients is really exciting for all of us, obviously.
So building off what you just said, Louise, I have like 6 different questions I want to ask, so I'll try to triage them. The first is, there is a tremendous amount of optimism about what gene therapy can do. And one thing I've heard repeatedly from researchers in the field is that patients, and particularly their parents, when we're talking about pediatric patients, are so eager to catch the gene therapy wave that they're not willing to participate in other types of research, enzyme replacement therapies or what have you, that may actually be more promising at this point in time for certain diseases. Do any of you have thoughts on that? Do you want to comment? Is this something that you see, or is this not something that you think is a factor?
Maybe I'll just start quickly. I think one thing that we've seen is that because of all the activity in the field, that patients do have options, which is fantastic, because years ago there wasn't anything. So the fact that they have options for their physicians to speak to them about is great. And that's all we can say, is to speak to your physician about each and every option and make the best decision for you. So that's, again, optimism that there's options, but yeah, there has to be a balanced description of all the various options. Terry, BJ.
Yeah, I guess I'd say I could certainly understand that perspective, but I was actually at a meeting in Europe a couple of weeks ago and heard a presentation by the parent of a child with a metabolic disease, for which there's a fairly effective small molecule therapy. And his view was, given that that therapy is very effective, he's not at all inclined to be going into gene therapy. He clearly understands the risks and benefits. And I know it kind of made me think. I think there's a lot of parents that can understand these situations and make these decisions, I think, appropriately.
Yeah,
Thanks, BJ. And I'm gonna be picking up on that in the hour. So hold that thought, and we're going to come back to it. Go ahead, Terry.
well, I think PJ said, my experience with families is that they do want to try multiple different options, and I think that one of the more likely scenarios that is what I think Louise may be referring to is sometimes, even though there are several thousand, probably as many as 7,000 different single gene disorders that could potentially be addressed with gene therapy, it seems that certain genetic disorders have multiple different products sort of competing for enrolling patients at the same time. And I think one of the concerns that families have is that currently it's quite difficult. For instance, if one were to have participated in one AAV gene therapy trial and received a certain AAV, perhaps it ends up being a lower dose or a less ultimately, once the data comes back, a less effective treatment, that may preclude the patient from participating in other trials. So that's a very peculiar thing. I see some uneven distribution of the trials and products that are being offered. In other words, sometimes there are multiple trials open at the same time in one genetic disease and many other genetic diseases for which there are none. So that does to some extent relate to the biology of the different targets and so forth. But still, I know it's very frustrating for families.
I'll just press you on that for 1 second. You don't really have to say anything further, but I'll just say I've noticed that unevenness too. And as you said, to some extent, it's due to the plausibility of the biology involved in that particular target. But it seems in other cases, as one would expect from commercial entities, what are being targeted is the less rare of some of these rare diseases, which makes me think that it's, financial considerations of what's the pool for potential recipients down the road that's driving innovation. And we've also seen, at least in Europe, the withdrawal of a gene therapy product because there wasn't a commercial base out there to support it. And we're starting to see in some other areas, once you've treated the population, suddenly there's not a commercial demand for a product anymore. So anyone want to delve into those treacherous waters of adding commercial realities into all of this?
Sure. I'll jump in. I mean, I think that's a big reason why we developed, when I say we, a large group of people have focused on the Bespoke Gene Therapy Consortium. It's really that issue, the realization that there are many diseases that could certainly benefit from gene therapy, but the ones that get taken into clinical trials the fastest are those, the most common ones. for understandable reasons. And you have these other ones though, where the plausibility is equally as good, if not better, and they're not getting in there. So a big focus of the Bespoke Gene Therapy Consortium, which is an effort supported by the Foundation for the NIH and involves NIH groups working with private industry and patient advocates, is to try to streamline the whole process of starting up AAV clinical trials, make it more standardized and more efficient, with the idea that that may, first of all, change what we think about as a disease that is a commercial interest, if we can kind of make it simpler to start up a trial, and really to help out some of these groups whose diseases are so rare that they're not going to get the commercial interest at the present time. You know, and as I think we know, Terry knows, we've all seen these families that ultimately sort of have to create their own small gene therapy companies. And it's a difficult situation, but I think that's a big focus of the Bespoke Gene Therapy Consortium and also the PGT program at NIH are to really help these diseases that have no commercial interest.
And Dr. Brooks, I think we're probably, I could be wrong, I'm not up to date, a few months out from y'all announcing what indications you're going to be targeting. Is that true, or did y'all already announce that?
No, you're right about that. And it's where it kind of gets into what I was saying. When for the clinical part of the BGTC, we ultimately want to choose up to 6 or so diseases to do AAV clinical trials, really for the sake of having sort of example diseases to streamline the process. And so there's a big question, a very difficult question about what are those 6 diseases. And the way we decided to make that decision is first have an open call for sort of proposals. We got about 61. We've able to cut that down now to a group of about 14 or so, and we've asked for subsequent information, and then from that we'll be cutting it down to up to, I think we're anticipating 6. But that just kind of illustrates, you know, that those 61 all had pretty plausible cases for AAV gene therapy trials. You know, animal data, stuff like that. So there's a lot, and it's not our goal to cover all those. Our goal is to streamline the process, but there's clearly a need out there. Absolutely.
Which, you know, I should have mentioned earlier when I talked about the sort of scientific progress of bringing new gene therapy products to market. You know, that's also being matched by regulatory innovation, by people thinking about like the ethics and the policy and the commercial implications. All of this, it needs to be proceeding hand in hand, or else what we'll end up with is products that are not available to the people who need them. So
yeah, and I would be remiss if I didn't point out the critical role of Peter Marks and CBER in getting the BGTC stood up. It was really in great part his vision. And of course, he's well aware of these regulatory hurdles, and his interest in this, I think, made the whole effort possible. Without the active involvement of FDA and CBER, you know, the big companies, they don't really have a lot of need for NIH grants. But the thing that links us all together, all the stakeholders, we can all benefit from a more efficient regulatory process.
Terry, Louise, anyone want to jump in?
Just add a few comments, and I think I agree with everything PJ said, and I think the Bespoke Consortium is wonderful, and it does take all of us to work together. I think one thing in industry, we're certainly interested in treating rare diseases as well, and one thing that we've been able to do is utilize our platform. So we work on all rare diseases, but some are rare, some are ultra rare, but they're related diseases like limb-girdle muscular dystrophy, for example. And so we're leveraging the biology, the technology, so we can also treat those ultra rare diseases as part of our platform. So I think it's going to take all of us working together to get there, but we're just as interested as everyone in kind of getting to these areas. So it's an exciting time to see everyone working together to get there.
Yeah, I mean, one thing that you just mentioned that I just want to pick up on is, you know, sometimes we have a name for a disease. It is disease X, but there's subtypes within it. And, you know, those subtypes may vary in prevalence. So even within a disease, you have discrepancies in terms of how rare it may necessarily be for a particular patient. Terry, did I cut you off? Go ahead.
No, that's okay. I didn't want to step on your your clarification here, but I feel like perhaps it's my role here. I'll go ahead and put a plug in for academia, but also take some responsibility from the side of academia. We do, of course, do go where the NIH leads us in terms of grants. But I will say that in my observation, part of the unevenness of the distribution of clinical trials is because certain key investigators have published some sort of, excellent, proof of concept studies and or taken things, very early, first in man types of trials, which then really de-risks to some extent, the corporate investment if it's gone far in academia and there's know-how and so forth. So I think that I do think it is important for the academic community to stay grounded, not just in what are the easiest targets for gene therapy, but in our responsibility to patients and families that are affected by the various diseases that we encounter. So I do think it's great that the community is. I feel that there is a great spirit of cooperation, and we have had great leadership at FDA, in CBER, and in NIH. I think PJ, also the previous, the immediate past NIH director, of course, Francis Collins, very strong advocate for this, and PJ himself and others within NIH. I probably skipped some people I should mention, but I think that was wonderful. Those two programs he mentioned, the PGT and the BGTC, have really called out an agenda of having these prototype diseases that are rare single gene diseases that are amenable to gene therapy. And I also think the NIH has, before we kind of get off of that, it's been done a really good job with this trying to drive the gene editing. The somatic cell gene editing consortium has been very productive, I think, and really help move things forward in a way that kind of helps level out between, where industry, doubles down and attracts investment to develop cures based on specific intellectual property. I think sometimes the NIH funded programs done through academia can develop, new things, or can kind of flesh out in a little bit less IP, driven way the applications to particular diseases of things like new technologies like somatic cell gene editing, for instance.
You said that academia tends to follow where NIH points the way, with regard to funding opportunities and whatnot. But I do also want to note that particularly in rare disease, it's pretty common at this point to have patient advocacy funded projects as well, at least more so than in other therapeutic indications that I'm aware of. When I talk with patient advocacy groups and ask what their playbook is, very high on the list is find a researcher who's working on something relevant to this disease and then see what we can do to assist with their efforts. So I don't know if anyone has any comments on that.
I mean, that's undoubtedly true. And I think, one of the first, I believe the first use of AAV9 for gene therapy in humans was in giant axonal neuropathy. And much of the funding to get that to the clinical trial stage was from a patient advocacy group. And then the clinical trial was done at the NIH Clinical Center. So I do think that in some cases, these patient groups have been leading the way. In some ways, where I feel like what we're trying to do is kind of help them out. And that's one of the goals of these programs.
And especially as you point out for the ultra rare, where there may not even be a patient advocacy group, or if so, what it's able to fund is probably quite limited.
Yes.
I want to throw out another question for whomever wants it, and it just a very global question again, which is, I asked earlier what you were excited about. I'm now going to flip it and say, what's the biggest concern you have about the current state of affairs or where you see things moving in the near future.
I guess for me, it's kind of the same issue that we've been talking about. It's all those diseases that aren't going to get to their clinical trials where the proof of concept and the biological plausibility is so clear. And what do we do to help them? But I think that's what we've just been discussing. So I think that's one of the biggest challenges I'm seeing in the field. And, we've been talking about what we're trying to do about that.
I think I'll just elaborate one dimension of that is that, we get contacted, and I'm sure Louise and PJ get contacted as well, from parents whose child has a progressive disease. And so they understand they have a limited window. And so if we say, well, yeah, your disease will probably, get to a trial in 5 or 7 years, that maybe never for their own child. And it's heartbreaking, quite honestly, when you have to relay that that's the current state of affairs. So yeah, that is it's that kind of in-betweenness of it where, when I was in training, not even that, you don't even have to go back that far, but when I was in training, we would say, oh, well, it's these genetic diseases are really fascinating, but unfortunately you can't do anything for them. And that it's not quite. The parents see the very promising results with some genetic diseases and say, well, why can't, what's happened for type 1 SMA happen for my child? It's very hard reality to deal with.
I agree with everything that's said. Maybe just to take a slightly different angle on it. As far as, I don't see any major challenges. I just see things where the field is evolving over the next 5 years. And what I think you'll see is continued improvements in manufacturing, getting higher yields to be able to potentially, bring things down, make things faster. Also just realizing where you can use a platform and where you can't. And so I think there's a lot of things we can leverage from platform, from AAV, but just knowing what you can leverage and what is different from disease indication to disease indication, because there may be some differences in the way a particular vector works in a certain disease. So kind of having a more defined path for those, I think we'll see that evolution over the next few years. So that's where I see things going.
Yeah, and I think that the question about leveraging platforms, that's a lot of what we think about with the BGTC and PGT and gene editing, and is a lot of what we're trying to explore. And can we just narrow down that path? Because it seems in principle straightforward. If you can deliver a gene to some tissue using a particular serotype of AAV, and you only swap out the therapeutic gene, what it should go, the whole process should go faster, and that's what we're trying to explore. So yeah, I think finding out that pathway, but I think even more broadly, what I often wonder about is whether it would ever be possible to commercialize a therapeutic platform as opposed to commercialize treatments for one disease at a time. It seems like scientifically it should be, but when I talk to people about this, it doesn't seem that way. So that, I think, is something that maybe there's an opportunity there in the future.
Well, thank you all for answering that question. And I'm going to give myself the privilege of sharing what my biggest concern is. And it shouldn't be any surprise that as an ethicist, I come down to equity. And to me, I'm concerned about equity on two fronts. And I would love to hear your thoughts on either of these. One is the cost of the approved product. At this point in time, we're still trying to figure out how payers are going to be able to afford, massively expensive products, particularly things like Medicaid, where there's a, set budget that needs to be stretched across everything. And one unanticipated expense of $2.3 million is going to wreck the entire budget. So I'm worried about, both within the United States and internationally, how people are going to afford these enormously expensive products, even though, the argument can be made that perhaps they save money in the long run by diminishing the amount of costs that need to be spent to care for a patient. So that's the one aspect of equity that I'm worried about. The second is, and I'm no scientist, I'm an ethicist, but I have heard from people that there is a concern that if we put all our eggs in, say, like an AAV9 basket, what we're doing is building in inequity from the start because prevalences of these viruses vary globally. So a virus that may be usable in a certain context may end up being not as usable in another area of the world where that virus is naturally more prevalent. So I'm going to stop talking and ask if y'all want to take on either of those two things.
There's a lot to.
Yeah.
Oh, go ahead.
No, go ahead. Please go, Terry.
Well, I just want to point out that there are some efforts going on to deal with the global equity issue. ASGCT, there's a committee that's dealing with sort of global outreach and trying to get to the middle income countries to expand availability of certain gene therapy products that might be able to be applied in those settings. I also think that when you look at the cost of gene therapy, the model that I have tried to impress on people is you could think about conditions which could be treated with a bone marrow transplant. And, there again, you have a very large upfront cost, or I should say stem cell therapy transplant, because it's often umbilical cord blood now or mobilized peripheral blood. But the point is that there's a big upfront cost that saves down the line a variety of morbidity, mortality, and costs. And so, there is no question it's a big upfront cost. But if it, I know that there are models where that is looked at in more of a process spread out over a number of years, at least as a total expenditure. But I do think that this is not easy. There's not a quick answer to this question of global equity. And so I think it will take a lot of work over a long period of time to gradually disseminate some of these products to where patients might be, whether that be, for instance, thalassemia has a big global prevalence, other hemoglobinopathy, sickle cell, of course, so these are things that really demand an effort for global access.
I just want to note for anyone who's not familiar with the acronym, Terry, what does ASGCT stand for?
American Society of Gene and Cell Therapy. There are also industry efforts to do this as well. So I don't want to slight them entirely, but I don't want to mention one and not others.
PJ, were you trying to get in?
Yeah, I think I could answer this. One thing we think a lot about, particularly from the standpoint of gene editing, but also we've heard this now about delivering messenger RNAs using nanoparticles. You can imagine those would be a lot less expensive to produce. And, one of the benefits of this horrible pandemic is the realization of using messenger RNA delivered in nanoparticles as a vaccine. But you can do that to deliver messenger RNAs encoding enzymes in a kind of gene therapy. I mean, to me, what's the most attractive and intriguing is to deliver genome editors as the messenger RNAs in those nanoparticles along with the guide RNAs. Something we focused a lot about in supporting this kind of research as part of the NIH somatic cell genome program, and we'll be doing some more of that. I think when you get to those kind of ideas, nanoparticles being a lot less expensive, having to give them once if it's a genome editor, under optimal conditions, it seems to me that would reduce the price and perhaps even allow some more dissemination of this technologies into other parts of the world where you don't have access to the kind of productive facilities for AAV or even for ex vivo gene therapy, for example. You have to take the hematopoietic stem cells out. If you could, in vivo delivery of genome editors in vivo using nanoparticles is something that we're very excited about for a lot of reasons, including the equity and issues you mentioned, Alison.
Great, PJ, that's wonderful. Louise, did you want to comment?
Yeah, I just wanted to comment on the one topic that we didn't touch on yet that you raised, which was about the prevalence of AAV antibodies globally. And certainly, I think with some vectors, this has been seen. With others, not as much differences in different areas of the world. But one thing I'll mention that is that we and almost every single gene therapy company in academia is working on ways to remove antibodies. So whether it's treating patients with pre-existing antibodies or the potential for redosing. So I think with all of those efforts that will hopefully alleviate that, along with improvements in manufacturing so that we can get to areas of the globe that are less fortunate.
I was actually going to ask specifically about redosing. Would you be willing to tell us, anyone, about what's happening with that? Does that look like something that we're going to be able to do at some point in the near future, or anyone's guess?
Yeah, I can touch on it. So we certainly have several different approaches on the horizon, both preclinically and then clinically. Tackling first patients with pre-existing antibodies. The antibody titer that you have after gene therapy is much higher, and we've shown that we can do it preclinically, but you want to start with the patients with pre-existing antibodies first because they're lower, and demonstrate safety and proof of concept there, and then move to redosing. But certainly in large animal models, it's proved to be safe and efficacious. So we'll start clinical studies soon, and I think the field's definitely going there. So it's another area where people have rose to the challenge to help find a solution.
I know when I've spoken to parents of children with these diseases that want to participate in gene therapy trials, and sometimes they are told that that's not possible because of the pre-existing antibodies. People have said things along the lines of, it's like the emotional impact of the diagnosis all over again. It's just like this devastating, no. So I'm really excited to hear that there is actual, a light at the end of that tunnel, perhaps, that these people will not be precluded forever. Anyone else have anything else they want to say about redosing? Then let's move on. The one thing that nobody brought up as a challenge that is in our Q&A several times is safety. So, the recent high-profile death of a particular patient has been mentioned. And so, Dr. Flotte, I didn't know if you wanted to just announce to everyone when the press conference will be with more information about that. And then let's talk about safety in general, whether it's insertational oncogenesis or whatever it is. How big of a problem is safety for this field?
Yeah, so I think you're referring to the tragic death of a patient in an N-of-1 trial that was sponsored by the Cure Rare Disease organization. A very brave young man that I had the privilege to know who had Duchenne muscular dystrophy. And as the family and CRD, which is a family run foundation, shared in their post, this patient passed away, heroically deciding to participate in this trial. And at the family's request, additional information is not being shared until it really is all gathered through a multi-institutional, multidisciplinary group. And the Muscular Dystrophy Association is sponsoring a conference in the latter half of February to publicly discuss different aspects of that. What I would say broadly about safety is that it's quite important to look at whether in individual cases, whether you're dealing with individual situations or with a whole class effect of, PJ has been talking about the platform. So, is something related to every patient who gets a certain serotype of AAV at a certain dose, or is it related to a particular cargo in an AAV, or is it related to something else, or is it particular to a disease, or even more so to an individual patient? I mean, there was a high-profile announcement with the BioMarin hemophilia A product of a patient that developed a B-cell malignancy recently. And I think the sense is that was an individual scenario. We also, another example would be that in X-linked MTM trial from a couple of years ago, it turned out there was a toxicity that was really related to underlying disease. So I think it's fairly important, and we now have the good fortune, and it's as a field to have, thousands of patients participating in these trials, and so that the patterns can emerge about what some common toxicities are, and these have been more discussed more publicly. And there are some AAV specific, and there are some retro and lentivirus specific dose limiting toxicities or vector platform specific risks. And I would just say that it might sound trite to say this, but with every particular decision of whether or not to move forward with a given product or with a given individual patient, it's always a risk balance or risk benefit calculation. And so the field has the potential, by sharing data, to work to identify predictable risks and improve the technology. And I certainly think that the Duchenne field is a very great example of that, where that has been happening. Identifying at-risk patients, identifying dose limitations on safety and so forth. So I guess that's kind of the key, is ends of one never really are fully informative, but pooling of information is very informative. And so I just say, in that context, it's very important not to overinterpret a single result.
So can I ask just a naive question? I should know this. I know that serious adverse events in trials must be reported to the FDA, but is there any sort of public reporting of these so that other scientists, other people in the field, are aware of what has happened and can be on the lookout or try to avoid these?
Yeah, we are required. We report these publicly in scientific meetings in addition to the FDA. So certainly it is important. And I think Terry summed it up well, but it's always a risk, a benefit. And I think we're now treating hundreds, in the realm of hundreds of patients. And so the more information you have, the more you can just say what is specific and not. So there are differences between AAV serotypes and disease indications, certain patient profiles, and so we're learning more with each patient that we treat.
I want to push my questions just a little bit further, because Louise, I would imagine for companies, they're like SEC requirements to divulge these so that shareholders know what's happening. But Terry, for academia, is there a requirement for public adverse event reporting or non-publicly traded companies?
Yeah. I mean, there is I would say there is, there's no longer reporting to, since there is not like an NIH RAC or equivalent, there are various reporting requirements. In general, the sponsor, which is often a company, not always, can be an academic institution, has to do the reporting to the FDA, and the principal investigator, the academic investigator, in most instances, has to report to their IRB and their biosafety committee and just any local authorities. But right now, there's not a general public reporting requirement. But I believe that they've more or less all of the information comes out. The trouble is sometimes it kind of dribbles out, but I would just contend that there's often a real push from IRBs and other places to certainly make this information available to other participants in the trial or potential participants in the trial. And as soon as that happens, this gets posted on social media in various places. So unfortunately, there's not as much sort of normalization of the quality of that, but the disease foundations also are often very helpful. For instance, they, like in the case of the X-linked MTM, they disseminated some information, early, and then the company did later more information, but not all the information was available at the same time. And then ultimately, one of the investigators gave some very excellent, scientific presentations after the whole picture kind of came together. So I think that's kind of the way the field is going. So it's not completely cut and dried like that, but the information does come out for sure.
Can I just, for those who aren't familiar with it, you mentioned the RAC. Can you explain what that is and why it doesn't exist anymore?
Well, I'm not sure I can explain all of why it doesn't exist anymore. I might have to lean on PJ a little bit for that. But the RAC is was the recombinant DNA advisory committee that originally came out from something that was originally called the Office of Recombinant DNA Activities, and then the Office of Biotechnology Activities. The RAC was actually an entity that was set up at NIH in the late 80s, actually maybe the mid 80s, when they were originally to look at plasmids, when human genes were being put in bacterial plasmids. And then when it got into human gene therapy in the late, I think 89 was when the first, Steve Rosenberg did the first tumor infiltrating lymphocyte marking studies, and then Mike Blaze did the first severe combined immune deficiency patients at NIH. Then RAC started doing individual protocol review and putting out kind of total, reporting of all kinds of different information. But it turned out that RAC really didn't have any. They were advisory to the NIH director. They didn't really have actual regulatory authority like FDA did. So they were kind of left in this funny scenario where everybody asked people to go to the RAC, but its role was kind of redundant, and it wasn't constituted the way that FDA expert reviewers are. So I don't know. That's my kind of probably somewhat butchered up history of RACs coming and going, but I
thought that was pretty good, considering I sprung that question
me too.
on you with no advance warning. PJ, anything you want to add to that?
No, I think that's about right. I think the way Terry explained it, and there was a time when every human gene therapy trial had to go to the RAC, and then I think there was a decision made that at some point that was no longer necessary, unless I do think there's oversight if you're doing something particularly new technology that involves impacting the genome. But I think the days where every AAV gene therapy trial had to go to the RAC are no more, and which I think is reasonable.
So I want to ask one more question about safety before we turn to the audience submitted questions. And that is, I've talked with some of you in the past about just the inherent failability of animal models for some of these rare genetic diseases. And I was wondering if some of you would like to take up that issue and how that applies to our current state of affairs of research.
Yeah, I guess my thought is that clearly you need animal testing, but I think we've got to start really asking what is it that some of these animal toxicology studies are telling us, because they're very expensive, in many cases using non-human primates, which are being a depleted resource. And the reality is, despite all of this very rigorous, very expensive testing, we're seeing a lot of serious adverse events in humans. And so I think it's worth asking, and it's one of the things that we're going to try to ask in the BGTC is, can we identify sort of a minimal set of preclinical studies, animal toxicology studies, that would be sufficient to assess if something is safe to give to humans. Very importantly, I want to emphasize our goal here is not to, cut back to the point where we're risking safety, but really asking, what can we find out? What are these studies telling us? And carrying out animal studies, expensive animal studies that aren't giving us information that are protecting patients, it is just wasteful. So I think it's an evolving area, certainly, but I think one that I hope will continue to evolve.
Louise, I saw you nodding. Do you want to
Yeah.
add anything?
Yeah, I would agree with that. And, it also comes down to the models. Does a wild type model tell you the same thing as a disease model? And I think we've seen differences there. And I think, if the consortium could come to a point where there's standards for the manufacturing procedure that's allowed, then that could also limit what needs to be done. So you don't need a tox study to show you that your product is pure enough, for example. So I think it's all going to co-evolve in terms of the CMC requirements and toxicology potentially. So there's a lot of work being done there.
Yeah, I do. One of the other things we hope to get with the BGTC is a limited and agreed upon set of critical quality attributes for assessing AAV vector preparations before they go into humans. And I do think that does get into has impacts on how we consider safety.
Terry, any other thoughts on safety or animal models or anything before we move on? And let me encourage people to be using this time to drop questions in the Q&A.
I don't really have any more to add. I think you've summarized it really well. It adds to the hurdle, adds to the activation barrier to bring a gene therapy forward when you have to do redundant animal studies, and it adds to the financial hurdle.
So I want to note that not only do we have an expert panel, we have expert audience members as well. So we have people who are updating us about the state of affairs of the RAC and the FDA requirement for academic investigators and pharma companies with regard to serious adverse events. So if you're not looking at the Q&A, I refer you to those helpful comments. There is one question. We touched on this earlier, but I want to circle back to it because we have a question from an attendee. The question is, would anyone care to address the public health implications of the high cost of these gene therapy products? So I know we talked about cost earlier, but anyone want to specifically take on that question?
Well, I just want to emphasize, maybe this relates to also the individualized applications of a platform technology. Things that are products get highly regulated, highly scrutinized. There are people who do things in academic medicine that are very expensive, surgeries and other procedures that are not regulated in the same way. And so I think that we have to look at the potential for lifelong benefit. So, I mean, I would certainly love for it to be less expensive, but I also think cost will come down over time. Real cost, inflation adjusted cost will come down over time.
I agree with you that I believe costs will come down over time, although I'm not an economist, I'm an ethicist. But I just wanted to say, when PGTME began, the first thing we did is we had listening sessions with patients, parents, other stakeholders. And I still very vividly remember the comment from the mother of a SMA type 1 patient who said, my insurance company paid for Zolgensma. I'm incredibly thankful that they did. And yes, that's likely decreased, the sort of lifetime expenses that my child would have incurred. However, my child is still in a wheelchair, still needs specialized food. And now they're denying these claims because they're saying we gave you the cure. So I think that, as all this works out, we need to figure out, when we're talking about value pricing, et cetera, does that truly, wipe the slate of other costs, or are there still other costs that need to be covered, and how do we accommodate that so that, we really are meeting the needs of patients and their families? That's an editorial comment. That's not a question. Anyone want to say anything further on pricing before we move on? Well, we're in the last.
Alison, it may be trite for me to say this, but I have to say it because it's the way the families that we work with feel. It is somewhat, difficult to put a price on, given the families hope and an opportunity for a child to live a longer life, or even for an adult to have have a better quality of life. And I just want to say all therapies are very expensive now. Your therapy for psoriasis, your therapy for arthritis, your therapy for this, nobody's developing cheap treatments very much now. That's not where the biopharmaceutical field is going. So let's keep working at it and get the price down.
Yeah, and I'll
No
just,
more,
I
no
guess,
more 15 cents for your small molecule, whatever. Yeah. PJ?
yeah, again, as I said before, I do often think that if we could somehow get these things to become therapeutic platforms, so every time you wanted to do a new disease, you weren't starting from scratch, I think that would change things. And I will put a plug in. We are trying to fund trials of that in the somatic cell genome editing program, basically requiring people to do more than one disease at a time from the start to see what that regulatory path looks like. Because not being an economist, it does somehow make sense that if we got to that point, things would be a lot cheaper.
So we only have 3 minutes left. Time has flown. I want to ask our panelists if they have any, pressing thoughts or concerns that we haven't touched on yet that they want to share with the group. I'll tell you the one thing I wanted to get to that we didn't get to was talking about the follow-up for trial participants. The FDA requirement for a multi-year follow-up and what is needed structurally for that to happen, because companies go out of business or they're acquired by other companies or whatever, and how are we going to get that long-term data? But we didn't get there. So Terry, I'll ask you.
It's a great question, Alison.
Thanks, PJ. Terry, I'll ask you to go first. Any last point you want to make for our audience today?
Well, no, I just thank you for holding this forum. I think it's wonderful to see that. I think people who are in this field are optimists, and they see the fantastic potential of this, and getting people together to share that enthusiasm is a really good thing. So thank you for doing that, Alison.
Well, thank you for being a part of it, Terry. PJ, any last comments from you?
No, I think we've hit all the major issues. We've certainly touched on safety. We've also touched on the recent approvals. We've seen some really great clinical benefits. We're starting to see that for gene therapy, and I hope that will continue.
You know what? I want to apologize. I suddenly just got a whole host of questions in the Q&A that hadn't been there up until now. So I want you to know I did not intentionally ignore your questions. I just didn't see it. Shoot, that makes me sad. Maybe we'll try to address some of these in the rest of the week, since we do have 4 more events coming up every day, noon to 1. But Louise, any last comments from you in our final minute?
No, I think everyone said it well. I think, again, there's a lot of reason to be optimistic, and I think now it's time for the community collectively to work together, continue to be diligent about efficacy, safety, continue to innovate in manufacturing and safety, and I think we'll be in a tremendous place 5 years from now. But thank you for having us. I think this is a great forum.
Well, again, thank you all for participating. So with that, we're out of time. I want to thank our panelists for a great conversation. I enjoyed it, at least, and learned things. And I want to thank the attendees for their questions. And I really am sorry we didn't get to all of them, but I'll share them with the other moderators for the other events this week and see if we can get some of them addressed for you. Be sure to tune in for tomorrow's panel. You can see on your screen that it is about unrealistic expectations and decision-making support, which I personally think is a huge topic for our field. So on behalf of PGTME, I would like to thank all of you who attended, participated, helped plan, or in any other way were involved in this day's event or the lecture series. You can see Kara just dropped our planning committee members' names in the chat. So a huge thank you to them. And I hope you all have a wonderful rest of your day and hope to see you back here tomorrow. Thank you so much.
Thank you.
Thank you.